It's an honor to be here. I'm very flattered to have been asked, especially to talk about something that I think is really super duper important, and also happens to be the closest thing that I have to scholarship remaining in my declining years. So I wonder if I have do I have co-host power? Can I share my screen?
Yes, I think everybody should be able to share a screen, but I also made you a co-host.
Oh, you're the best. Now I feel super special. Okay, so I'm going to share my screen, assuming, oh God, this is where I always mumble to myself as I do this, so my students know that I'm old. I'm going to stop that screen share because it was the wrong one. It's also unnerving to share your screen and you hope you don't have that really inappropriate tab open. Okay, I think I'm safe.
Yep.
So, can everyone see that okay?
Yep.
Excellent. So in 1997, I had just finished up, I think, the first 6 months of my postdoc at The Ohio State University at the Rothenbuehler Honeybee Lab. And I had submitted an NIH, well, I'd submitted a grant to the NIMH for my work with honeybees. I'd also simultaneously submitted a grant to a now defunct foundation called the Xeric Foundation, which was founded by Peter Laird, who was one of the creators of Teenage Mutant Ninja Turtles. And Laird had created this foundation to help self-publishing cartoonists, comic book artists at the time. Because at the time, the world of alternative comics was just sort of growing, and unless you did superheroes, you weren't going to find much support at publishers. And I had in mind this idea for a comic book about the life cycle or the life of a honeybee called Clan Apis. So I submitted those grants at about the same time. I found out at about the same time that I received 3 years of funding for my postdoctoral research, salary, supplies, all that stuff. I also received a $1,500 grant to publish my first comic. And I will tell you, the thing that was most exciting was the $1,500 grant. That's the thing I was like, hey, honey, guess what? So the idea of storytelling in science has always been important to me, and I've been trying to work on it for several years. All right, so I'm going to show you some books as I go along, simply because they have been valuable reference material for someone who is not trained academically as a storyteller. The first is A Storytelling Animal by Jonathan Gutschall. This is a terrific book. And perhaps one of my favorite quotes pulled from it, evolution is ruthlessly utilitarian. So how has the seeming luxury of fiction not been eliminated from human life? Fiction, storytelling, that frankly takes time away from hunting and collecting and finding mates and fighting and sleeping. Why would that persist? And that's sort of one of the theses of the book as he goes plugging along, looking at how it is, in fact, that we are critters that are profoundly wired to tell stories. Anyone who has had children knows this. To receive stories, right? We love listening to stories. And we love it so much that what are some of the things we miss during this pandemic? Well, I miss going to a giant room and watching stories with complete strangers. There is a natural communal participatory nature to storytelling that makes it profoundly compelling. And what I'd like to talk about today is sort of capturing that compelling nature of storytelling to convey the information that we want to in a class, right? To make it sound totally utilitarian. I hope to soften it a little bit as I go along there, but that's sort of the big idea for the day. So why do we still tell stories? in part, we can't help ourselves. So I want you to take a look at the kitty cat in the box here. I've turned down the volume. You can see so here's a cat. Looks like it's just sort of doing cat things. Something's bothering it. It's hopping around. It's pretty remarkable, right? It's really going after something. That cat's asleep. It's in full-on REM sleep. And it is part of the work done by a researcher known as Juvet. And Juvet, he actually found the part of the brain in the cat, and we have a corresponding part in our mammalian brains as well, that is responsible for paralyzing us in sleep. So that's interesting, I think. Paralyzed in sleep. So most dreams aren't dreamy, right? Most dreams aren't happy. They're not always nightmares, but they're almost always designed to help us or deal with a problem. So there's some problem. It could be absurd, like we don't have pants for class, or it could be something as simple. My wife Lisa told me about a story about her concern about chapped lips and how to handle those chapped lips this morning. These are the things that we dream about. We dream about solving problems. So that cat looked like it was going after a mouse or something. It was dreaming about a problem, an issue it deals with. But because this region of the brain was disabled, it can actually physically act that out. And there are individuals that have disorders in which they do physically act out dreams, and doing so can be dangerous, right? They could hurt themselves, they could hurt others. So when evolution, and I'll anthropomorphize here, please understand, I don't really believe that evolution has a thoughtful agency. When evolution had to decide between selecting against dreaming and the potential damage it could do, and setting up an entire complex wiring system to paralyze us during dreaming, evolution picked paralysis. And so there must be something profoundly important about it, that we would tell ourselves stories and act out stories in our dreams. The compulsion to layer stories over things for which there technically isn't a story is also pretty overwhelming, and we can't help ourselves. So take a look at this, this animation, stop motion animation done by Hyder and Simmel in 1944. When people are shown this, they are absolutely and utterly compelled to create a narrative. Typically, individuals who see it complain or talk about the big bullying triangle, the smaller triangle defending the little circle who's afraid, and the little circle is going to go inside and hide, close a door. None of that's happening. Those are just geometric shapes that are being moved around on a flat surface, and we're taking pictures of them and stringing those pictures in together. But we are developing and giving those things agency. We're giving them life, and we are constructing meaning from their movements. We cannot help ourselves but to tell stories. Our brains desperately, desperately want them. And it's unfortunate that we have for so long ignored that compulsion in teaching. It was not the nature of my teaching or the teaching that I received, even though I had some terrific instructors. And in fact, storytelling was routinely considered to be something that, well, was not very objective and somewhat subjective. And I don't know how many times I've heard people talk to me about dumbing down the material. My response to dumbing down is, if your students actually understand it, you're smarting up. I don't know if that's a term. I just made it up. Okay. So our brains are wired for it. We're compelled to do it. So what do stories give us? I typically don't like made up those portmanteaus that are clever and cute, but I did read one once on the back of a book about genetics called Wonderstanding. And I think that it captures essentially what I try to do in a lot of the work that I do, which is to say that, displaying facts just aren't enough, right? Making a comic book version of a textbook, a richly illustrated textbook, isn't enough. I think there needs to be stories, and I think that stories do two really important things. Well, one really important thing, and that is that they inspire. Uh, Again, I have a very specific opinion of what my teaching should be, and that is that it is not solely the conveyance of content. It is as much about inspiring interest and wonder in my students as giving them, the fundamental steps of the central dogma, or talking about how an action potential is conveyed down an axon. And the reason why is that when you inspire wonder, you draw students into material. If I feel wonder, I may in fact be motivated to, at base level, do the actual reading for class because I'm somewhat interested. At the best outcome, to start doing reading other than what I assigned to do because I find it interesting. We don't really ever see interviews with famous scientists who talk about stumbling across an exhaustive list of the characteristics of metazoans and being just totally inspired into becoming a biologist. It's usually some interaction with nature, some interaction with the world, and that interaction typically has a narrative structure. This thing inspired them to want to know more. So that kind of wonder is highly motivating, and I think stories do it astoundingly well. The other thing stories do is that they help forge relationships. So if you ever see a picture, National Geographic, anywhere, in a movie of a storyteller, they are typically not sitting by themselves. They are typically surrounded by individuals in rapt attention. When you tell stories, you build bonds with your listener. You develop relationships. And the research shows pretty clearly that those types of relationships are really important for underrepresented groups and marginalized groups. The feeling that they are connected to their faculty, to the other story listeners, is critical for setting up sort of this ongoing relationship with a student that is going to help them succeed. When I think about the students who I've not been able to reach, who I've lost contact with, the ones that don't succeed are the ones who just shut down. They go away. They're not part, and that's hard. And so what I try to do is set up a situation where I can develop those relationships so that I can more easily intervene and make contact. So I think that when we think about stories and we think about wonderstanding, we think about the idea that you're going to learn something, but you're also going to be inspired by that thing. And that inspiration is going to drive your motivation. And it's also going to help you forge relationships with the people that are going to help you succeed. Great. If you have questions and they pop up in the chat and you want to interrupt me, I have a deep and abiding love for my own voice, and so I will just keep rolling on. You let me know. So Dan, if you are checking the chat and you want to intervene, you please do so.
Will do.
All right. So there's a body of work out here. I've even contributed to that body of work that suggests that storytelling can be an effective way to draw both scientists and non-scientists into our understanding of the world. So I've taught non-majors classes. I had one non-majors class called sensory biology, and I got an NSF grant. It was an old CCLI, I think they're called IUs now, that to develop a comic book textbook. The premise behind this comic book textbook was it followed Wrinkles the Wonder Brain, and Wrinkles was the lab assistants for the Greya sisters, the three sisters from the Perseus legend, who are actually scientists, not witches. But he's in charge of carrying their eye around to them in the lab, the one eye that they share, and he drops it into a vat of distilled human imagination. In order to retrieve it, he has to go on a series of eye-related adventures. I know that sounds super exciting. And so there are about, I can't remember, 8 short little comic stories in which, so for example, on the left here, we have a page from the chapter on phylogenetics and the evolution of the eye, in which he's talking to Yggdrasil, the Norse, legendary Norse world tree that can take on any form of any tree, and in this case does so as a phylogeny. In those short comic stories, we have an adventure and a character, and we have distilled into it, or instilled into it, I should say, basic scientific concept. But of course, not a ton, because you've got to have room for story as well. And so what we do is, or what the design of the book was to show some story, introduce a few ideas, and then interleaving these comic book stories were more traditional text book type of pages. I think usually 3 to 4 that I would write, in which I would take the ideas introduced and I would expand upon them in a different way, maybe a little more traditional way. So the comic or the story allowed them to dip their little pitties into the water of science, and then you jump into the textbook. It's a little closer to the deep end. We tested it, it worked, and it seems as if when we looked at attitude surveys, we looked at their opinion of science and opinions of comics, and especially for the non-scientists, because there's a ceiling effect with the science majors. The non-scientists had a dramatic increase or significant increase in their overall opinion or improved opinion of biology, which correlated with their opinion of comics and story. So it seemed as if this method could draw in students who, at least in my experience, were traditionally resistant to talking about science, who had routinely told me that they can't do science, even though most of the kids were getting, I would have kids that have A's coming into my office hours saying, I just can't do science. I'm like, well, we're not actually doing science, we're talking about science. But that said, you are doing pretty well. They're all essay tests, so you seem to be able to convey the information. But they're steadfastly convinced they can't do it. I think stories help us overcome some of those, that resistance, that challenge. And you know, there's a model of success, motivation that I sort of keep in my mind, and it has to do. It comes from a book called I think it's called How Do We Learn? I can't remember. It's usually on my shelf, but the model involves 3 parts: efficacy, value, and support. And the idea is that if all 3 of those things are aligned, then a student will be motivated. So if they feel a sense of efficacy, that can they do the work? If they see value in that, in the work, in the studying, oh my gosh, what was it? And support, if they feel supported in their endeavors. Well, I think stories do all of those things. I think that a storytelling gives a kid a sense of the value. It's accessible, and so I think they have a sense of efficacy in terms of understanding the material. And I think that just the nature of storytelling transmits at least the suggestion of support, which should obviously be backed up with actual support. But it sends a message that I care and I'm trying, and I want you to understand this. All right, so it works. We published on it. It was exciting. Now, I typically, as a speaker, love to talk about the theory, but as a listener, I much prefer to see the practical applications. So I am going to summarize the academic literature on the benefits of storytelling in teaching. Okay, there we go. People like them. Also, they work. You can easily find, there are good scholarly articles out there that make it absolutely clear, and we've known for a long time that if you convey information in a story, people learn the information faster, they remember it longer, and they remember it better. And then there are other studies that talk about, moving up Bloom's taxonomy. So what I just told you right there was sort of knowledge and understanding. If you have those, then students that have those in a solid way are more effective at the analysis, as you move up Bloom's taxonomy. And there are papers that demonstrate that as well. All right. So we're going to continue for the rest of my time here, and we're going to talk about how you could possibly do this yourself, because I know that's the big question. To do that, I'm going to talk about something that's super duper fresh. I haven't talked to anybody about this. We just submitted an IU's grant to further develop this concept. But we have sort of overhauled our general biology course at Juniata. So when I first came here, general biology was taught by one person in the first semester and second semester of the student's career. We had usually 420 kids per class that came to Juniata, and half of them were in the biology class, whether they wanted to be biologists, doctors, or had to take it for their environmental science and studies. So we would have 200 kids, which for a small liberal arts class, institution is not good. Over the years, we've sort of whittled that down. Most of my time here, it has been 2 classes of about 100, a little less. This year, as my big initiative for chair, and I'm raking in the big bucks of $500 or something a year. It's not worth it. So when you have the power, use your power. And what I wanted to do was transform the way we did things. Now, obviously, I couldn't do this if I didn't have buy-in from my department, and I am beyond lucky to work with a group of people who are terrific science education innovators. And when I brought it up, there was really no struggle at all. I think people were ready for trying something new. So as it is, the structure of our course now is we offer it in both semesters. In the fall, we offer it for biology students. In the spring, we offer it for the environmental science students because that fits better for their sequence. And then in each semester, we have multiple sections. And so in the fall, we have about 4 or 5 sections, in the spring, 3 or 4, and generally class size is between 15 and 20, which is fantastic. Now, that's sort of the logistical size thing. It may or may not be critical, but I think it's important, especially if you're going to try to get to know your students pretty well. But let's talk about turning our general biology class into a sort of a story-based curriculum. What did we want to do? Well, the first thing we needed to do was we needed to identify critical concepts that we felt the students should have exposure to in Biology 1. And so as we were doing this, we used NSF's Vision and Change as a guide, our own personal experiences with biology. We looked at other people's syllabi from other institutions. We generated a list. Then we voted on priority, what was at the top of the list. This list, I don't think is the actual prioritized list. I couldn't find it, but the top 2 were definitely the top 2, central dogma and natural selection. So we generated this list, a list we could all agree upon for the most part as being important. We knew that we couldn't do everything, but we were going to see how much we could do within this context. Okay, so then the next step that we took was that we wanted to identify some cool stories in biology, right? I should say before I move on, right? Central dogma, natural selection, energy transformation, homeostasis, a lot of these map to vision and change as ideas and skills that students should have coming out of biology. So our idea is aligned pretty well and could be nested within a number of the ideas that NSF had outlined. All right. So then what I asked everyone to do is draw upon their expertise, right? So everybody comes into their own teaching and their own classes, and you guys know the cool stories. That's what makes faculty so much more valuable than, say, someone who's a non-expert in an area coming in and maybe working through the material from the textbook in a highly competent way. But be honest, when you're talking about what makes a class go kapow, that's when you've got a faculty member who says, oh, by the way, let me tell you the backstory on this set of research. Or we're not going to talk about this in class, but this is related to this, which is super cool and currently going on, right? Those are the things that nab you. So one of the stories that you might already be using in class that, or stories that you've heard that, man, I wish I could work this into a class. These were all things that we sat down to do. And of course, I work with, as I said, fantastic faculty who are really super imaginative. So we came up with a list of different stories. And then from that, we picked what we thought would be the best, the top 5. We were going to break things down into 3-week chunks, just arbitrarily chosen in order to get through the semester. Okay, so our resulting list of stories were some pretty cool stories. So photosynthetic slugs. I don't know if you're familiar with Alesia chlorotica, but we'll talk about it in a little bit. That'll be our guiding example because we just finished that one. That's the introductory one. So these are slugs that are capable of consuming a chloroplast and incorporating it into their tissues, and then the chloroplast continues to work until the organism dies. The evolution of speed. So what makes a cheetah so fast? And why is a cheetah fast and why isn't a lion by comparison? What are the structural functional elements that have to change? And so we follow a cheetah, and a cheetah becomes our sort of our, I don't want to say spirit animal, but our focus. And we talk about all the adaptations that have come about, and that spins off into various physiological adaptations, all the while talking about what's happening to that animal in the split seconds that it's running, and how the antelope or the Thompson's gazelle it's chasing, how it's changing, what it's dealing with. It's also fast. And so, so we do that. The opioid crisis focuses on, well, the opioid crisis, but it does so by looking at how opioids work. And I'll talk, well, I won't talk a lot about it, but the opioid crisis was sort of the brainchild of Jill Keeney, who is molecular biologist, well, trained as an immunologist. And so it focuses on how the opioid receptor is built. You got the central dogma there, how receptors work. We get some neurobiology in there. And then we also pull in, with the opioid crisis and the other 2 stories, we start pulling in social relevance. And I'll talk about that near the end. But with the opioid crisis, one of the most compelling things that we did was talk about racial inequity in terms of access to medicine, and racial inequity, frankly, in how the media depicts addiction and how we treat addicts depending upon their race. Alligator ecotoxicology, that was suggested by our ecotoxicologist who did work on an alligator situation in Florida in which toxic waste was dumped into a lake and it turned all the alligators into females. And then finally, cholera and climate change. This is by our microbiologist, Gina Lamandella. And it talks about Rita Colwell's work with, i.e., first of all, identifying a dormant form of cholera that lives on an interesting little copepod. And then how cholera outbreaks are exacerbated by climate change, anthropogenic climate change. So those were our 5 stories. And they worked as stories because of the next step. So the next thing we did was we needed to take those stories and see if we could map some of the basic ideas that we said were important to a given story. So again, we're going to stick with photosynthetic slugs. Here's Alesia chlorotica. I think I teach invert bio. I think it's absolutely gorgeous. So then we mapped these ideas to the photosynthetic slug stories. And we did this for all the other stories, but I'm not going to belabor each one. With the photosynthetic slugs, what did we see? We saw energy transformations being something we could easily do. Genetic transmission, structure and function, life histories. Now, the truth is we could have done a natural selection in central dogma. But we knew that natural selection was going to pop up in the evolution of speed. The central dogma was going to be in the opioid crisis. Um, and that this particular story lent itself particularly well to energy transformation, photosynthesis, also to genetic transmission and structure and function life histories, as we'll see. So we mapped the concepts that we thought would work in that particular story. And then we sat down and we essentially wrote a short story for each one. I'm going to read you the one that we did for photosynthesis, because I can get it all on one slide. That's the other key. We had to keep it short. The story itself had to be short and focused. Okay, so here it is. As a juvenile, the slug Elysia chlorotica extracts chloroplast from the algae Vaucheria latoria and incorporates its chloroplast into its tissues. All right, so now with this topic, we can do symbiosis, endosymbiosis. We talk about the differences between eukaryotic and prokaryotic cells, obviously. The slug never has to eat again because the chloroplasts continue to function. So here we got photosynthesis, energy transformation, until the slug dies 10 months later. This is possible because the slug carries genes critical to chloroplast function. So here we talk about horizontal gene transmission that they pass on to their baby slugs, vertical gene transmission. Although the chloroplasts make G3P for both slugs and algae, how the G3P is used differs between the two organisms. Here we pull in the life histories of these two animals. Slugs make comparatively more glucose. This is in large part because they're mobile and they have determinate growth. So they're moving around and they have to find mates, they have to find food, and they have to avoid being eaten. So they have more glucose than the algae, and the algae uses the G3P to make more structural molecules. So that's because this particular critter is sedentary and it has indeterminate growth. So in other words, it continues to grow and grow and grow and grow. Whereas the determinate growth of the slug means that it's going to stop getting big at some point, right? They do not grow indefinitely. You have determinate growth. I used to have determinate growth until I started gaining weight, and so it's now sort of a vertical determinate growth. Anyway, interestingly enough, the slugs all die simultaneously at 11 months old. This is a brain bender. They live for about 11 months, and it doesn't matter whether you keep them in captivity or you're in the wild, whether you've kept them in captivity by themselves or with others, they all died about 11 months. There is evidence that they picked up a virus from the chloroplasts whose viral DNA has been incorporated into the slug's genome. So again, horizontal gene transmission. A surge in viral particles seems to correspond with the mass deaths of the slug. So this last bit, this is, I didn't put it in here, this is scientific process, and it's put in because we actually don't know exactly what's going on here, depending upon who you read on this topic. And so we end naturally with, well, we start early in the life of the slug and end with the death. That's what animals and plants are really nice, also microorganisms, because they have a sort of a natural built-in story arc. They're born, they live, they typically try to do the same thing, which is to survive, make copies, and not be eaten, and then they die. So you have, in terms of the storytelling setup, a very similar thing. I imagine you could have the same thing with a chemical reaction, before, during, and after, right? So you could find that arc, the hero's journey, in virtually any type of thing that occurs over time, I suspect. Okay, so that's the story. That was how we go about building it. It took us a long time. So we spent 2 workshop days all day long working out both the topics. Well, I should say, I posted topics. We voted on them well in advance of the actual workshopping. We sat down and workshopped what we thought was important. Then we also workshopped the stories. And at the end of those 2 days, we felt like we were pretty close to having a skeleton that we could then flesh out. And then each individual, we took the stories and we broke them into groups so that individual teams could sort of develop the materials for that story. And they did that over, piecemeal over the summer. And then when we started in the fall, we were completely unprepared. Despite all that work, we thought we were great. And then we realized, oh my God, we're not ready. And so there was still a fair bit of scrambling despite all that pre-work. So last semester was a bit stressful, but teaching it this semester is actually really great because most of the materials are set up and now we're tweaking and refining and making things a little bit better, I hope. So as you're thinking about this and you're saying, well, okay, I think I'm going to try this. The thing is, what you can do is you don't have to develop a whole course, obviously. You could say, well, I've got to, if I were in neurobiology or animal behavior, I've got to think about reproductive behavior. I'm going to pick one animal, reproductive behavior of, oh, I don't know, something. And I'm going to structure a story that may last one period, it may last a week, it may last 2 weeks to get the content in that you're interested in. But you can use that particular story to illustrate various things. Now, as you're thinking about story, and Dan mentioned this book, Houston, We Have a Narrative, there are a number of different elements that Randy Olson outlines in the book, and he gives you sort of essentially templates and worksheets to sort of bang things out and to identify the basic structure of the story. It's really worth reading because what he does is he shows you that scientists have been using storytelling techniques, effective scientists have been using storytelling techniques for, well, most of their careers. He highlights, regardless of what you think of Watson and Crick, he highlights their paper, which is very short, but has the structure, the storytelling structure that I'm about to describe to you here. One of the things Olson points out, and he got this particular idea from the guys that Trey Parker, who does South Park, of all things. This structure that's known as the ABT structure of a narrative. It's the and, but, therefore structure. And if you look at most good stories, in most good science articles, especially abstracts. So in my Talk Nerdy class, we actually analyze abstracts for the ABT, and the really good ones have it nice, neat, and clearly. The ABT structure says that we have some buildup, some foundational stuff, this and this and this, right? And so I'll tell you the ABT I developed before writing a graphic novel about Santiago Ramon and Cajal, which I just finished actually, and I'm very excited about that. So once there was a little boy in Spain who loved nature and wanted to be an artist, okay? But his father said he had to be a doctor, bum, bum, bum. So the but presents the conflict. And that conflict can be, actual adversarial situation, or it can be something we don't know, right? We know this and this and this, but we don't know how this happens, right? And the but sets up the last phase, the therefore, where you start talking about what the hero did to solve the problem, or the heroine did to solve the problem, or what the scientist did to solve the problem, whatever the case. It's a very natural structure and one that I think most of our brains slot into pretty easily. Therefore, he figured out how to be both an artist and a doctor and won the 1906 Nobel Prize in Medicine. So Cajal was essentially a founder of modern neuroscience who found a way to manipulate the Golgi stain in such a way that he could identify and clearly define the fact that the nervous system was not just one great big net of cells, but in fact, individual cells that were talking to each other. And his art literally is hung in exhibits, traveling exhibit, I think that was sponsored by MIT. So once you have a story, whether it's for a day, a week, 3 weeks, see if you can't set up this basic structure to do it. The and can be this, laying down the basic facts. The but, can be, well, whatever the but is. Now, when you do this, it sounds, when I'm saying storytelling, like you're just going to sit up there and you're going to, kick back in a rocking chair and put a pipe in your mouth and say, hey, let me tell you a story. In reality, there's micro lecturing. All right, there are times when you're lecturing. For us, so we have little bits of where we set information up. A lot of the times within the story, we'll give them data from papers and say, okay, work through this data. How do we explain this element of the story? How do we explain what's going on here? And then, the students will come back and report out. And so in that way, we're actually all contributing to the story, moving it forward. So that is the way in which you can structure sort of within a particular day or a week. He also points out that stories are episodic. And most of us have watched or binged watched a show on Netflix, multiple episodes, somewhat consternation when they don't come out all at once. But within any story, there's a rising prologue, and then there's a repetition within that story of episodic events, say the trials of Hercules. Those map pretty well to the posing of a question and then hypothesis, experiment, results, hypothesis, experiments, results, et cetera, et cetera. And when scientists, by the way, report their results, they also use a story structure, the same one, IMRAD, right? There's an introduction, that's the and. Materials and results. All right, the introduction usually has the and the but, puts up the problem. The results, sort of the episodic adventure, here's what we did, here's what we got, and then the therefore is the sort of results and discussion. You can do the same thing with a class. Each class could be an episode. This is what our slug story looks like, so we mapped it out. Now, this is just the internal document. It doesn't look very story-ish. This is just to remind instructors of what we're doing on each day. But each day, we remind kids, we touch base with the slugs, we remind them of their life cycle, we remind them of where we are in a particular story. Here's the thing that you have to be prepared for in stories. If you're going to tell a story, if you're going to, now I suspect this is a crew of highly enlightened educators, and so probably we're not all hardcore lecture only, that there's a fair bit of problem-based learning. But usually you have to give up some content because you've got to make room for that story. You got to make room for the story to give you several things that I think make stories useful. The first is context. This is one of the things the National Science Board is routinely in their reports complained about, is that science is oftentimes taught without a lack of coherence. Well, a good story is coherent, right? A good story provides context. Context and coherence provide a scaffolding for that information. And once you scaffold, whether it's through a mnemonic or through a plot device, suddenly it becomes a lot easier to remember things. And this to me is something I try to impress upon my students, is that the remembering stuff is important. That is the base of Bloom's taxonomy. The firmer you understand stuff, the less working memory you have to consume holding it in place. I have internalized the English language. I do not have to constantly translate or think of the words I want to think of as I go through. Therefore, I've freed up working memory to deal with the problem at hand, right? That is what story helps us do. That's what context and coherence helps us do. Characters make you care. They make you care about, and whether it's cartoon insects or real life scientists, they're a way in for students to connect to the material. And then at some point I put comedy on here because I like to tell jokes, but this is a critical component, the ability to be light, to see the humor in things, to look at a problem sideways, and not to be afraid. I think on a cartoon earlier, I have a cartoon of a bee leaving a flower, and it says, "Thank you for transmitting my gametes to my lover." And the bee says, "Don't make it weird, man." It is weird, right? That's weird, but it's, in my opinion, delightfully weird. Or maybe that's coming up. I don't know. Another thing to keep in mind when you tell stories and you construct stories, and this was told to me by a junior adult librarian. There are two really important elements to a story. The first is the mirror, and the second is the window. So with the mirror, you're reflecting something. Your story is reflecting something back at the reader or the listener that they can appreciate. For us, we've made the mirror in our stories a human connection that is socially relevant or deals with the scientific method as performed by the actual people. So within the photosynthetic slugs, we take some time to talk about Lynn Margulis and the endosymbiotic theory. And more than just talk about her coming up with it, some of the resistance that she faced. And, was that resistance because it was a kooky idea? Because it did sound kind of kooky. Or was it a kooky idea because a woman came up with it? These are the pressures that sometimes women face when they, and I speak as a white man, so I hope I'm not sounding, inappropriate, but according to my colleagues, this is something, a problem that individuals face in academia. With the opioid crisis, we have the students read an article by Helena Hansen and Jules Netherlands on the war on drugs, and we focus a lot, because the article does, on the social and racial inequities that are surrounding the opioid crisis. As just one example from that discussion, we talk about how so many of the headlines were the new face of opioid addiction, or the new face of addiction. And when we did this, I had, we don't have a hugely diverse a campus, but I had 4 students of color in the class. And I asked the class, well, what was that former face? What's implied there? And of course, they all pointed out very quickly that what they're saying is the old face of addiction is brown or black. The new face is white. And this article points out that the way in which, white people are considered addicts is that they're victims, right? Victims of someone else oftentimes who introduced them to drugs. Whereas underrepresented minorities tend to be weak or criminals, right? This particular story resonated extraordinarily well with the students. And in fact, it's the first time I've been nervous walking into class for a very long time, because this is going to be really thorny and difficult. But we actually wound up hearing from admissions who were doing sort of focus group studies. They didn't specifically ask about anyone's classes, but a number of kids volunteered how much they appreciated this discussion. Another one, we talk about cholera and climate change. We talk about Rita Colwell and Colwell's ghosts. So when Colwell suggested that there was a dormant form of cholera that might live on copepods, the CDC sort of mocked her and called them Colwell's ghosts. As Colwell later said, now it's in the textbooks. And we talk about, in that particular story, access to water and sanitation, how it's not fair and unequally distributed, and then how further, climate change, anthropogenic climate change generated primarily by industrialized countries, are ultimately having a trickle-down effect to the countries that don't have access to easy water. And the rising sea levels allow the copepods carrying in the cholera to infect water supplies, and without proper sanitation, you're going to see blossoms of cholera. Right? So that's the mirror. The window is this opportunity to see a world that's completely unexpected. So like in young adult literature, that's Narnia. In biology class, it's kleptomaniac slugs that can photosynthesize. And having an opportunity to show kids that things aren't super well defined as they think they might be, that the line between insect and animal and plant isn't all that clear. The way in which sex is determined, and consequently gender, isn't necessarily clear-cut biology, as some would suggest in the media. It opens up the questions, and it makes biology, at least, less about stamp collecting, as Rutherford called it, and more about open-ended story problem solving. All right, so I'm going to close with getting started, because sometimes you don't know where to go. And so let me just summarize and suggest some things here, if this is interesting to you. So whether you're doing this alone or as a group, you should list what's important to you, the ideas that you want your class to do, or a section of your class. And then you should go hunting for inspiration. Your own experience should come first, but there are sites like the Next Gen Science Standards Phenomena page, that's linked, they got the link here, that has a whole bunch of different videos that could be quite inspiring to you, which could in turn inspire stories. Whittle down your stories, map those concepts to the stories, and then find a way to structure smaller stories per lecture. Again, focusing on the human element or on side bits of the biology of the organism. They can be stories about biology or people. And as I said, organisms have a natural story arc. So biologists have it relatively cool and easy. And the last thing to be prepared for is the process is iterative. You're going to go through this over and over again. You're constantly going to refine. You're going to have to trim. Then you may add back every once in a while. But what you'll find is I think that trimming and giving yourself space to tell a story that's compelling is going to make your students better. And it's going to help them be more enriched. It's going to make them better citizens if they're not going to be scientists. And I think in general could be quite a fruitful way for you to teach. So I think that's all I have to say. How'd I do? Whoa.
Done, Jay.
I pushed it right to the edge, just like in class.
We do have a few questions. I'll ask the first because a couple others had the same question. Somebody asked about assessment. So what is biology doing in terms of assessment to see whether the curricular revision is better than what they had before, or maybe even is benefiting those from historically underrepresented groups in STEM?
Sure. So this is a great question. What we're going to do, a couple things. We are assessing what we've done initially for the grant and what we're going to use as our baseline is persistence in the POE and an academic gap. So Dan, you and Ursula did some tremendous work for various HHMI proposals and sort of generating data about the existence of an academic gap. And it's clear, there's an academic gap between our white students and our underrepresented minorities, right? That they don't score as well. And that is actually clear too in our own biology classes. We're also looking at first gen students as well. So as we analyzed the data for the grant, what we found was after doing this for one semester and having one semester's worth of data for this class, we actually closed the first gen gap, the academic achievement gap for first gen. We did not close it for underrepresented minorities. It narrowed a little bit, but it was not significantly different. Well, it was significantly different. So we're going to look at that academic gap, but we're also going to look at persistence because what we know is that when we're looking at underrepresented minorities, they tend not to leave the institutions they're at. They tend to move to other disciplines, social sciences, et cetera, et cetera. And what they oftentimes report is the relevance and value is clearer to them. And so this is one of the reasons why we think it's important to make room for socially relevant stories every once in a while. So we're going to also be looking at persistence in the major from freshman to sophomore year, but also through graduation. And then finally, we're going to have, we've developed with Kathy Westcott an attitude survey on social, words slipping me now. Engagement, social engagement. And that survey is really going to sort of get to the idea of biology having a role in social engagement and whether or not, if we look at pre-post test with freshmen who are coming in their first year, whether after being in the class, their ideas about social relevance or the social relevance in biology has shifted. So right now, those are the primary metrics that we're looking at.
That's great. Thanks, Jay. We have maybe time for one more, and I'll ask it because I think some others are probably thinking this as well, particularly those at large, let's say R1s with high enrollments. What do you say to folks who say, I just can't do it. I can't cut out the content. I can't find the time to create stories. What is your response to folks who say that?
So I say this with all the respect in the world. There is no way in hell you can teach them everything you know. So I'll just say it like this. We quite successfully produce lots of MDs and PhDs. And in fact, small colleges, I think per capita produce more PhDs. We'll have to talk to Vince Bonacorsi, but I think that's true. So clearly At smaller institutions where this approach tends to be the case, we're doing okay. We are preparing the students well enough. And the truth is that in my neurobiology class, I use a lot of story-based stuff and not like sit around again. It's this sort of problem-solving kind of stuff. I get through a third maybe of my textbook, but I hear from students all the time that tell me that they're way ahead of their colleagues in medical school, because if you pick the right things to focus on, and this is the problem, textbooks tend to be all encompassing. It's impossible to convey all that information. You will have to make a choice. And if you choose well, and sometimes that's going to take iterations over decades, you can prepare your students extraordinarily well so that they know a handful of things so well that they can think laterally in a very effective way.
Yeah, that's really helpful. Thanks, Jay. I'll back you up on that too, which is to say, my approach is, and I'm not going to say how I run my class. Some folks know, but I also use sort of, you can call it a storytelling type of bend to it. And my take on it is, if students are only going to retain 10% of the stuff, and by telling story, it bumps that up to 20% long term, I'm talking 6 months after they leave my class, then is it really being a disadvantage to them if they're going to dump it all at the end of the semester anyway? Why not have a compelling story that they might be able to carry things with them outside of that class and beyond, right?
That is the key, in my opinion. Yeah.
So, yeah, very good. All right. Well, thank you so much, Jay.